So, yeah, thanks very much for the introduction. So, so this is a theme that's run through quite a bit of my work, and I thought I'd try and tie it together into a coherent story to present to you. So, we'll see how well I did at that. But the central idea is that there's a bunch of things that we normally think of as just being biological phenomena, which include growth, nutrient cycling, homeostatic regulation, food chains, and symbiosis. And I want to try and show you that these are all things that can just happen in abiotic systems that just have chemistry. And so from this, I kind of want to argue that ecology came before biology. So we shouldn't think of the origins of life as being about how the first cell kind of appeared on a dead planet, and then from that all the ecosystems, or what we think of as ecosystems, got created. Instead, we should think of it as being the ecosystems, meaning these sorts of things, as coming before the first cells, and in fact being the context in which they arose. So I'm not exactly the first person to make this kind of claim. These are some diagrams that I found from various places on the web, just kind of showing examples of how cycling can appear in non-equilibrium systems. So this one is a simulation of convection. So you're heating the bottom and cooling the top, which puts the system away from thermodynamic equilibrium. And as a result, you get this cycling of fluid. So this is another example of convection. But of course, in ecosystems, we also see a kind of cycling. So we see recycling of nutrients. You're probably familiar with the carbon cycle and the nitrogen cycle and so on. So this diagram is of the carbon cycle. And so it's been known for a long time that this kind of chemical cycling happens in non-equilibrium systems. And in fact, it's a theorem that if a physical system is held away from equilibrium, then you'll get cycling. And it's also being claimed that this can lead to growth, and that's what I'll spend quite a bit of the talk talking about how that can happen. So, a classic thing that you see studying ecosystems is this kind of curve of what happens over time when a species is introduced. So at first you get exponential growth, and then you get saturation. It sort of tends towards this carrying capacity, because at first you grow exponentially, but there's only so many individuals or so much biomass that the system can support. So basically, we're looking for can this sort of thing happen in chemical systems? And there's lots of reasons for being interested in that. One of them, again, I'm sort of assuming that most of you are sort of familiar with the idea of daisy well, because I'm not really going to explain it. And if you're not familiar, it doesn't matter, because it's not that important for the rest of the talk. But one reason to be interested in growth is that this type of argument, this daisy world argument, says that when you have a biological population interacting with its abiotic environment, it's often going to lead to kind of stability. It's often going to lead to, in fact, the conditions being maintained, like, for instance, at a temperature that's particularly good for life. And the assumptions that you find in this model, basically, you need that the organism grows in this kind of exponential fashion. It grows faster the more of it there is. And also that by growing, the organism has some kind of effect on its environment. And those are kind of really the only things you do need. And James and Ian published a nice paper recently kind of showing that if you just have random effects on the environment, you'll find this sort of self-regulation. So growth is the thing that we want to look for. It's the thing we want to explain. But of course, in a biological system, growth happens in a very, very, very complicated way. So these diagrams are just to give you the idea that it's complicated. So this is just the example of DNA replication, but I could have given you all sorts of other examples for vital processes from a living cell. DNA replication happens by a molecular machine moving along DNA molecules, like a tape recorder moving along a tape. And you're not really going to find that sort of thing just in a regular chemical system. So how can you get something like biological growth in a chemical system? So the term AutoCatalysis means something that catalyzes its own production. And so to catalyze something means to make it happen more rapidly. So if it makes its own production happen more rapidly, then that leads to exponential growth. And autocatalysis is very common in combustion chemistry, but there's only one abiotic example that's really known in liquid phase organic chemistry, which is called the Formose reaction, which can be used to create sugars. So the question then is, how easy is it to get autocatalysis with relatively simple organic molecules? So not things like proteins or RNA strands, but something much simpler. And there are several possible mechanisms. So I'm going to kind of run through some of them and then conclude that one of them is much easier than the others. So one of them is template replication, which is an idea that's very similar to the way DNA replicates, but without the enzymes. So here you've got this polymer that undergoes complementary base pairing, and then the two halves split away from each other, and so you started with one of these and you ended up with two. And although this is a bit of an aside from my main point, I want to talk about something I did a year or two ago now at Sussex University just before I left, where we actually built some template replicators, not on the chemical scale, but on the physical scale. So this is like an air hockey type table. It blows air up from the bottom, and these things are made of plastic. They're about that big, laser cut acrylic, and they have little embedded magnets. And the idea is that you can join two of these together and put them on this table, and then they will join other ones together. So I just run through it quickly because it's kind of fun. So the mechanism works like this. So you start with a string. This is AA. They can be joined together in AB, BA, and BB. And they can join, these magnets are opposite polarity, so they can kind of join together like that. Another one can come along, join in, and these are designed so that unless it comes in at exactly that angle, it's just going to bounce off. And then a collision can join those together, and then they split apart. And so this AA has made a BB, and then the BB will then create an AA, and it will have completed the replication. So this does work. There's me putting the thing on, and annoyingly, so yeah, annoyingly there's no way to skip that video forward for some reason. So I'm going to have to not show you the video, but it does work. You don't want to wait 4 minutes for it to happen. Oops. So yeah, it works, but the point of that is just to say that in order to make that work, I had to spend several weeks making these different plastic things and going, oh no, those ones don't work, because what happens is it's very, very, very hard to stop them from just coming together by themselves. So you end up with these kind of like crazy complicated things. And that kind of makes it a bit, or to my mind, kind of makes it a bit unlikely that that life is going to have begun that way, because then you need to have these quite complicated molecules to start with, and then you have to wonder, well, okay, but what process could have made those complicated molecules to start with? So there's another, just to quickly mention, there's another way of thinking about autocatalysis that's due to Stuart Kaufman. And he said, well, what if you have, he was thinking about proteins, so he's thinking about a scenario where you have peptide strings in quite high concentration, and so they're able to catalyze reactions between other peptides. And so he said, what if you had something like BA catalyzes the A plus B forming AB, and AB catalyzes A plus B forming BA. So they don't catalyze themselves, but they each catalyze each other's creation. And there's some very nice results from this, and there's an awful lot of work that that came from it. And so he basically showed that, yeah, if you make those assumptions, it's very easy to get these large sets of things that all catalyze each other. But you still need protein-like molecules. You still need quite complex molecules. So what I'm going to do is show you that there's another way to get autocatalysis. I mean, yeah, I'm going to show you there's another way to get autocatalysis. It's not that it isn't already known, but I'll show you a model that demonstrates it's also very easy to get that kind of autocatalysis. So just going to take another quick diversion to talk about experiments, because the aim of this exercise is that we're working on a kind of experimental implementation of these ideas. So we want to do something that's going to be vaguely plausible within the context of a real experiment. So this is famous, the Miller-Urey experiment, a very famous experiment from the 1950s. And what they did was they put these gases in this chamber, which what they thought at the time was the composition of the early Earth's atmosphere. Actually, it's now thought would have been slightly different, but it's a little bit beside the point. So the idea is that you put these gases into this chamber, and you also have this kind of simulated water cycle. You're boiling water here and it's condensing here. And you put energy into the system using this kind of electrical spark. And what happens when you do this is that it produces this huge variety of different products, including over 20 amino acids, which is great. It's fantastic. It creates all these molecules that make up life. Again, just to very briefly mention, there's another way to do something very similar, which is HCN hydrogen cyanide polymerization, which is what we're actually working on. And the way that works is you just basically get hydrogen cyanide in a reasonably high concentration, and these triple bonds don't really like to be triple bonds, so they undergo a series of reactions that convert them into single and double bonds, and that results in long, complicated molecules. And then you also end up with amino acids, among many other things. But the problem with these, or it's not necessarily a problem, but something you observe in these experiments is that you don't really get life. So you get these molecules, but they're all kind of jammed together and tangled up into this black sticky tar. And so the question, I suppose, one of the motivating questions of this is, how do you go beyond that? How can we, or could we possibly do an experiment where we might get these sorts of molecules, but they'll actually be doing something, they'll be kind of undergoing some kind of growth, rather than just all sticking together and then stopping. So how might we go about that? Well, one place to look is to think about what happens from a chemical point of view, from a kind of large scale, looking at it from a distance chemical point of view, when you have biological growth. So for example, if you imagine that we have this Petri dish with a sugar medium, and then we have some bacteria growing on it, the net reaction basically is you get sugar plus oxygen turns into complicated stuff, which then turns into carbon dioxide and water. So the point is that both the food and the eventual products are actually quite small molecules, and it's only in between when it's in the living state that they're long polymers. So in between you've got the building up of polymers and also the breaking down of polymers. So maybe catabolism is what's missing from the origins of life experiments. Maybe if we have something that breaks down the polymers as well as something that builds them up, then we'll get something interesting. And so what I'm going to try and show you now with a very simple, extremely simple model is that maybe once you've got those two processes, then you'll get autocatalytic growth, and then you can get kind of other more interesting things that look more like ecology. So this model it's extremely simple. So we just basically imagine a kind of well-mixed reactor containing oligomers, which just means short polymers, of various different lengths. And the point is that these are, unlike a lot of origins of life models, these are meant to represent quite simple molecules. These are kind of things based on simple carbon chains, like the kind of things you get from HCM polymerization. So they're not meant to be proteins. These things can't catalyze reactions. And the things that can happen are that these two molecules, if they collide, they might form a longer oligomer, or they might just spontaneously decide to split up into two shorter ones. So those are the only two types of reactions. So basically all the reactions look like this. Some string of some length plus another string of another length goes to a string of the combined length. And we just choose a random set of those possible reactions, which is, again, that's following on from what Kaufman does in his model. So it's kind of like fairly standard way to generate these artificial chemistry models. You just take, out of all the possible reactions, you take a random set. And that just leads to a set of ordinary differential equations. I haven't put them on the slide, but it's just a set of ordinary differential equations using pretty standard assumptions. And it might seem like, well, that might be too simple to get autocatalytic growth, but it's not. So I'm just going to show you a very like the simplest possible way it can happen. Actually, when you run the model, it happens in a more complicated way. But the simplest possible way it can happen is so if we imagine that we start this system with only monomers, so there's just monomers. But then now we kind of imagine that we put a small number of dimers. So dimers means an oligomer of length 2. Put a small number of dimers represented by this single molecule. And then, so we're sort of assuming here that the reaction, so there's a possible reaction which is just the two monomers come together to create a dimer. But we're assuming that reaction isn't allowed in this system. But maybe a reaction that is allowed is that dimer can come together with a monomer to produce a trimer with length 3. And maybe that can come together with another monomer to produce a tetramer. And then maybe it's allowed for the tetramer to split up into two dimers. So if we've got those reactions, then what we've done there is we couldn't have, before we added the dimer, we could never have got a dimer. It just wasn't allowed. But after we've added one, we've got two. And then those ones can then go on to create more dimers and more dimers and more dimers. So that's how you can get autocatalytic growth in this type of system. And that will continue until the supply of monomers starts to get used up, and then these reactions will start running in reverse, and it will come to equilibrium. So you can summarize it like that. And this is called an autocatalytic cycle. And yeah, these things are well known about. I just wanted to kind of show you it because it can be kind of hard to grasp otherwise. So these things are called autocatalytic cycles, and the point is that in thermodynamic equilibrium, all these reactions are balanced by their reverse reactions, and nothing really happens, which is just that's what thermodynamic equilibrium is like. But if you have monomers in excess, then the reactions are favored in a particular direction, and it leads to the cycle turning, doubling the concentration of dimers every time. So in this model, we choose the reaction set randomly, so we don't kind of force it to use that particular cycle. So we take this thing, we put this high concentration of monomers, we have these random set of reactions. So this initial state with just the monomers is far from thermodynamic equilibrium, just because of the way we set the parameters of the model. And what happens over time on a fairly typical run is something like this. So you can't see it, but at the very initial stage, what happened was that a large proportion of the monomers just almost instantly turned into dimers, because on this run, that reaction is allowed where two monomers can come into it, can become a dimer. But then for a long time, nothing really happened. And that's because there was actually autocatalysis happening, but everything was at such low concentrations that you just can't observe it. So this is typical of what happens if you have an autocatalytic reaction in real chemistry. You'll see it just sit there for perhaps quite a long time, and then suddenly you'll observe this exponential growth. And so that's what we get here, get this sudden period of exponential growth. And then after a while, you start to see, so I wish I'd made a slide with the actual cycle that you do get, because it's a more complicated looking cycle. It's actually not just a single cycle. But what happens after a while is that other reactions start to happen, because you've got all these different products, and all of these all of these molecules have a whole load of different reactions they can undergo, and so once they've built up to a certain level of concentration, they start undergoing all sorts of different reactions, and that's when the system really starts to go to equilibrium. So here you've actually got tar, essentially. So here you've got there's a lot of monomers and a few slightly less dimers and slightly less trimers than that, slightly less tetramers, and much longer polymers present in low concentrations, which is a sort of what tar looks like. But of course, you can hold the system away from thermodynamic equilibrium, and one way to do that is to put it in a flow reactor. So in this case, this is just a kind of simplified example of the simplified version of the model. But we have, in this reactor, we're continually adding monomers all the time, but we're also removing dimers from the system. And this is kind of the sort of thing that we'll be able to do in the real experiment, because you can just add monomers, you just literally just add them, and small non-monomer molecules can be removed by a semi-permeable membrane, because the larger molecules won't go through. So it's the sort of thing that we could do in the real experiment, but very abstracted. And in this case, we just allow all the possible reactions. And so what you see is you see this exponential growth, and then it's followed by saturation. So it starts to look like the kind of ecological growth that I showed originally. And if we take the system in this steady state, or the end of the run when it's almost in a steady state, what we see is this. And you can see that this is an autocatalytic cycle. So you've got A3 becomes A4, becomes A5, becomes A6, but then A6 splits up into two A3s. So every time you go around this cycle, you double the number of A3s. And if we do the same thing with n equals 7, then you get similar kinetics, so you still see this exponential growth and it's still followed by saturation. But it actually happens for a very different reason. So this is the reaction network that you see, and it looks quite different from this one. And so the reason is that this reaction, this A6 going to two A3s, is actually going the other way. So this has become a depleting reaction. And so this is just kind of interesting because it demonstrates this idea that the reactions, so in chemistry, in real chemistry, reactions can always have a forward and a reverse direction, and which way it goes depends on the concentrations. And so this is kind of interesting because it shows you that these cycles can kind of organize themselves by the system choosing the directions in which the reactions take place. There's a problem with autocatalytic cycles I haven't really talked about much, which is this problem of side reactions. I did mention it, that you have that all of these species can undergo more than one reaction, and so some reactions will contribute to the autocatalytic cycle, but others will just deplete it. And in this case, it seems to solve the problem by by being complicated. So it's very difficult to, so there's no reaction. This A7 can become A5, which isn't part of the autocatalytic cycle, except that it is, because it's just a different, it just skips over to a different part of the cycle. Yeah, it's hard to explain that without some other slides, I guess. So the point of this is that so autocatalytic cycles have been proposed before in terms of the origins of life. So in particular, Günther Wächter-Scheuser and Harold Morovitz have both talked about the reverse citric acid cycle. So this is a cycle that actually takes place in modern cells. So chemotrophic bacteria that feed on chemical energy, use this cycle to generate a lot of their component molecules. But in living systems, each of these steps has to be catalyzed by an enzyme. And so the question then, the debate was about, could this cycle happen if you didn't have enzymes to catalyze the reactions? So Wächter-Scheuser said that that maybe mineral surfaces could catalyze these reactions, and then other people said no, they couldn't, or they could, but they would also catalyze other things, and it wouldn't work. But this model or this research, maybe, hopefully I can demonstrate it experimentally. It should be a lot more convincing than the model. But it suggests that autocatalytic cycles are actually not so rare. It's not so hard to make them. So maybe, maybe the citric acid cycle is hard, but maybe there were other autocatalytic cycles that could have that could have preceded it in the origins of life. So that's the end of the first part of my talk, and I'll just briefly summarize what I've said so far. So we've seen how growth or autocatalysis can arise in abiotic systems, and it doesn't need complex protein-like molecules, just synthesis reactions and decomposition reactions, so just two things coming together and two things splitting apart. And that's because of this cycling theorem, that you always get cycling when you're far away from equilibrium, and then cycling itself can lead to autocatalytic growth. And that means it's probably not too hard to get autocatalysis in chemical systems. So, and as I've mentioned a couple of times, we're working towards demonstrating this in real chemistry. And basically the idea is to take this hydrogen cyanide polymerization idea, but also have hydrolysis reactions, which you can do just by changing the pH. And then so we'll have both this breaking up, the building up and breaking down happening simultaneously. And then the idea is that the mass spectra of these things are really complicated. It's very difficult to tell what's going on. But if we see sudden changes in the mass spectrum, then we can deduce that something interesting is happening, even if we don't know exactly what it is. But yeah, I'm not going to say a lot more about it than that, because this work is still at a very early stage. So in part two of my talk, I'm going to talk about some much earlier work that kind of comes from my PhD work. And this was about spatially self-organized chemical systems. And then I'll talk about how once you put these ecological features into these models, they do very interesting things. So this is a photograph of a chemical system that's happening in a sort of petri dish. It's an autocatalytic system, which and basically what's happening is it's not well mixed, and so that results in these kind of traveling wave phenomena. And I'll show you another example. So this is that was a real example of a reaction diffusion system, and there's something you can model mathematically pretty easily. So there are chemical reactions that happen on a surface, but as well as reacting, the molecules can also diffuse slowly across the surface. So this one has this reaction, U plus 2V goes to 3V, which is autocatalytic because you need the V, you need some V's in order to get any more V's. And this is, although it's a simple, unrealistic reaction, it's really just meant to stand in for the sorts of kinetics that you get from these complicated autocatalytic cycles. So the U here is the food, and this is fed in to every point in the system simultaneously. And you can get all sorts of different patterns. You can get these reproducing spot patterns that you can see here, but you can also get chaotic patterns and traveling waves and all sorts of things. And I'm just going to use it as an example to show how ecological feedbacks can affect self-organization. So what do I mean by feedbacks? So here's a cartoon picture of the biosphere. And so it's got organisms of various different types, and they're on the surface of the Earth, so they interact locally. But they're coupled to the atmosphere, which is which is kind of like a well-mixed reactor. So these things aren't well mixed; they only interact locally. But the atmosphere is pretty well mixed. So the mixing time in the atmosphere is about 1 year. But the residence times, meaning the time that a molecule of carbon or of oxygen or nitrogen will stay in the atmosphere, they're of the order 10 years for something like methane to 10 to the 6 years, which I think was for nitrogen. It was a while since I made this slide. So yeah, so things stay in the atmosphere for a long time, but the atmosphere is mixed on a fairly short timescale. And that gives rise to feedbacks because the gases in the atmosphere have effects. They're affected by the things in the system, and they have effects on the things in the system. So CO2, for example, is fertilizer, which means that if this guy makes some CO2, then these guys are going to be happy, even though they might be on the other side of the Earth. It's also a greenhouse gas, of course, and these things are sensitive to the temperature as well. And then similarly, there's O2. There's all sorts of feedback loops involving all sorts of different things. So what happens if we couple a reaction diffusion system to a kind of atmosphere? So we're taking this system with these local interactions, and we're coupling it to this kind of global feedback. I mean, this is so we're sort of assuming that in this case we're assuming that the food is fed into this atmosphere, this reservoir, which actually doesn't have to be the atmosphere. It could just be a pool of water or something. But we're assuming that food is being fed in at a constant rate, but the rate at which food comes out depends on how much food there is in the reservoir. And that just results in you get these equations, and they're coupled to an integral equation. You've got these partial differential equations, but they're coupled to an ODE, which represents the concentration of food in the atmosphere. Yeah. So then I show you the demo. So this G, so you can't see very much happening at the moment. So what's happening here is that the red stuff is the food, and this G represents the concentration of food in the reservoir. And what happens is, because of this feedback, that if I reset the system, then the G just goes up to a certain value and then stops. Because at this point the food is coming in and going out at the same rate. But now if I add a drop of AutoCatalyst into the system, then the first thing you see is that it grows and spreads out. But then you can see that this G starts to drop because the food is being used up by the stuff in the system. And pretty quickly, it found itself in this state where there were where you've got this kind of interesting patterns. And I'm not going to say so much about it, but there's a kind of interesting analogy between the kind of individualness of these spots and the individualness of living organisms. In this talk, I'm not going to talk too much about that, but I think it's really interesting. So I'll just show you that again. Because it happens quite quickly. So you add this thing, it grows, spreads out uncontrollably at first, but there's only a certain, it's limited. The amount of green stuff in the system is limited, and so it can't just keep on spreading out forever. It has to form into some kind of pattern. That pattern might just be one big blob, but quite often it isn't one big blob. Quite often it's something interesting. So, and this sort of seems to be a general result. What happens when you limit, so if you don't limit the amount of energy going into the system, if you don't limit the kind of population of the green stuff, then well, it can form patterns, but a very likely thing that will happen is that it just spreads out, and it's like having just a layer of self-reproducing slime across the whole Earth. It's not very interesting. But if you add these feedbacks, they tend to tune the system into the pattern-forming region of the parameter space. Now, how am I doing? Oh, now I don't know how I'm doing because I stopped the thing. Okay, so there's an explanation of why this happens, which I guess I'm going to run through, but pretty quickly. But it's quite similar to the daisy world sort of idea. So, so here we have, this tells you how the concentration of of food in the reservoir depends on the rate at which it's depleted by the green stuff. Get this curve. But there's also another curve, which is how the amount of green stuff in the system depends on the concentration of U. So, and it turns out just from the dynamics of the equations that there's a thresholding effect, where if the food concentration is too low, it just can't persist at all. It always drops down to 0. But if it's higher than a certain threshold, then it goes up to a certain value. Thanks. Okay, so I'm going to run through this very quickly. So yeah, and then this results in a steady state that if you set the feedback right, the steady state is actually just exactly on this threshold. And if you don't have the feedback, it's really impossible to get a steady state that's on the threshold at all. And so that was a schematic diagram, but this is my attempt to draw a real one. It's my attempt to calculate this actual curve, and it kind of looks similar to that. So let's skip over this bit. So yeah, I already described about the limiting the energy supply limits the amount of green stuff, and so if it's going to persist, it has to form into a pattern. And nutrient limitation has a similar effect. So if we assume that there's some kind of nutrient that is needed in order to build the AutoCatalyst, but that gets given back when the AutoCatalyst decays, that gives rise to very similar dynamics, which I'm not going to show you. So I want to talk in the last few minutes about food chains. And the idea here is that you can have these autocatalytic cycles which eat food that gets fed into the system. But you could also have another autocatalytic cycle that doesn't eat the food, but it eats the primary producer, if you will. And so if we add this just into the Gray-Scott system, it'd be nice to do this with the model from the first part of the talk, but I haven't done it. So we add this to the Gray-Scott system, and with the right parameters set just right, you end up with spots that have tails. So the tail, this green tail, is actually eating this pinkish spot, and the spot moves because it's being eaten on one side, and that just makes it move. So that's kind of interesting, and you can kind of make all sorts of interesting metaphors with that system. But something particularly interesting happens when we add in this feedback. So this is the new result from this stuff. Most of this part of the talk is old. So this is exactly the same system that I already showed you. So the feedback is tuning the system into the region where you get these spots. And an interesting thing is that a parasite, in order to persist in this system, has to be just right, because if the parasite's too fast, if it's too good, it just depletes the spot, and then there's nothing else for it to do, and it dies out. But I have a parasite that's just right. Whoops. So I've added it into the system. And now you can see these spots have kind of been infected with this parasitic tail, and they're moving around, all well and good. But they're actually doing quite well. They're multiplying quite quickly. And you can see these guys now aren't doing too well. And that's because this G value is becoming slightly lower, because these guys are actually, they're so good at reproducing that they're able to they're able to change this G value. And so these ones, even though they haven't been affected with the parasites, they're not reproducing anymore. And in fact, they're kind of unstable. And eventually you'll see them dissipate. There they go. So now we've got the system, and then this G value is 1.04-ish. And it turns out that in this system, the original spots without the tails, they no longer can persist. So now only spots with tails can persist. So that's quite interesting. And the reason that's interesting, or one reason it's interesting, so one reason it's interesting is just the fact that the parasite can be beneficial. So actually in the origins of life literature, there's a lot of people kind of worrying about parasitic reactions and whether they might cause terrible problems for AutoCatalysis. But this actually shows that they can be beneficial. So that's kind of interesting. But another thing that I think is really interesting about this is that you end up in a state where only the spots with the tails can survive, just the ones without can't. And so if we found this system, if this was a real system and we went there and looked at it, we might think, well, how on earth did those things appear in the first place? Because you need these two different chemicals in just the right relationship. If you just had the system and you tried to make something persist, it would be quite difficult unless you happened to put the green and the blue right next to each other with the right concentrations. But of course, we know that there's really this feedback. So it started out that in the beginning, persistence was very easy because there was lots of energy available. And so these two autocatalytic cycles can just arise one after the other. But the feedback creates this state where the original one can't persist anymore. So this is just a clue that maybe persistence was much easier in the past, and that's because of these global biogeochemical feedbacks. Maybe it wasn't so hard to make life to begin with. So it's just one more demo. I guess I've got okay. So it's just one more very quick demo, which just looks really cool. So the idea is to make so that we have two autocatalytic cycles, but they each produce a nutrient that the other one needs. So we've got this kind of the X. As it reproduces, it converts B into A, and the Y needs A and converts it into B as it reproduces. So intuitively, perhaps we might get red spots and blue spots. Perhaps they might sit next to each other because they want to, because they're exchanging nutrients. And it displays it on the wrong screen. Let's try doing it this way. Do you know how to make it not do that?
Make it not do what?
So when I double click on this, it shows it here, but not on there.
Mirror display.
Mirror.
So settings display. It might look a bit off.
Arrangement display.
So then gather windows. You click gather window.
But I've got a million windows open at my.
Underneath that. Arrangement. Mirror displays.
Ah, right. Okay, that's what I'm used to. So where was I? Right. So this orange bar is just the concentration in the reservoir. It's just an orange bar instead of a number. So I'm just restarting it so that I can explain it. So it starts out just kind of homogeneous. You've got the red and the blue just mixed together because there's so much food available that they can persist when they're mixed. But pretty soon it drops below the point where that can happen. So you kind of get a bifurcation where they become non-mixed, and then very, very shortly after that they start to form into spots. So here we've got, you might think of it as an ecosystem where you've got red spots and blue spots, and they're coexisting symbiotically. But as time goes on, this really interesting transition happens where they do actually start to sit next to each other, because this this blue will tend to move towards this red because it's where its nutrient is coming from, and vice versa. But they won't overlap because they compete for the same food. So, it's just interesting. It shows you that you can have this kind of symbiotic relationships, and it's just another piece of evidence that when you put these feedbacks into the system, it tends to tune the parameters to the kind of really interesting parts of parameter space. I think without putting the feedback in, I never could have found the parameter space where you see this pattern. So yeah, that summarizes what I just said. So you get this ecosystem of red and blue spots that are separate, but they exchange nutrients. But then it transitions into a state where you get symbiotic clusters of red and blue spots that live next to each other. So it's an example of feedback being useful for finding the interesting parameter ranges. And it goes some way towards demonstrating that this formation, so I briefly mentioned this idea of individuation, the formation of things that are analogous to living, to organisms, rather than just a homogeneous spread. So yeah, that's the end of part 2, and so now I just have a few slides to summarize before the end. So to summarize this last part of the talk, implementing ecological feedback seems to make these reaction diffusion systems more lifelike. And reaction diffusion patterns are not very much like cells at all. There's no molecular level processes, there's no DNA stuff going on, there isn't a membrane. They're isolate, they're kind of spatially isolated, but not through having a membrane. But they have in common with living cells that they're both autocatalytic chemical systems, and they both kind of somehow responsible for their own spatial separation from their environment. And so this research that I've just shown shows that it's just a first step in showing how large-scale feedbacks can assist in the formation of complex, lifelike, far from equilibrium structures. So a speculative picture of the origin of Earth might be that on the early Earth, well, we know for a fact that on the early Earth there are many sources of chemical-free energy. So these would include things like hydrothermal vents on the seafloor, but also photochemistry in the atmosphere would have given rise to something like the Miller-Urey experiment. So it produces this tar, and you can get energy from the tar. So in the absence of life to consume these sources of chemical energy, they could have actually built up to pretty high levels. So this is the idea that persistence is easy in the past because there's loads of energy around, there's loads of food, nobody's eating it. And this would make it easy for complex autocatalytic sets to persist without being bound in vesicles. So because these polymerization autocatalytic sets don't need such high concentrations of monomers. So they don't need to be bound in a vesicle. But if you do get the formation of a protocell, so it's autocatalysis surrounded by a membrane, which stops the constituents from diffusing away into the environment, then those would be much more efficient. And they would then drive down the available free energy in the environment through the types of feedback that I've spoken about. Until after that's happened, you would only find vesicles. You wouldn't find things just metabolizing in the environment without being surrounded by membranes. So yeah, summary and then conclusions and then the end. So to summarize the whole talk, energy flow leads to matter cycling in physical and chemical systems. Matter cycling can lead to autocatalytic growth, even in quite simple chemistry. Cycling and a limited energy supply can lead to these types of large-scale feedbacks. And the large-scale feedbacks interact with more local-scale feedbacks to produce this kind of spatial patterning. And these spatial patterns kind of have something in common with life. They look a bit like it. And so I've kind of tried to argue that, or at least I would like to argue that this type of process, this type of feedback, tuning the system could have been very important in the origins of life on the Earth. I haven't spoken much about evolution, but it's a big open question how you get evolution in these types of systems. I've got some ideas, but there's no time. So this conclusion slide summarizes what I already said, I guess, but these last 2 points are understanding the effects of these kind of feedbacks is important for understanding life's origins, and maybe understanding these purely chemical systems can help us to understand life or understand ecology.